My name is Marla Trindade. I am a professor at the University of the Western Cape. And just a very warm welcome to all of you. So today we're hosting this workshop, co-hosting with Diplomics today. But this falls under the banner of the BlueBiobankBiotech project and the Biodiversity Biobanks of South Africa, and I'll explain in a second how all of this comes together. But I actually like to start with thanks and acknowledgements first. So this is all the schools and the foundations that are represented here today. So welcome to you all. We're so delighted that you are participating in this. Then I just, on the right, I just want to thank the Diplomics team, to Shane and Tim. They've been excellent, wonderful supporters of my research group and our laboratory and the research that we conduct. We're very thankful to everything that you assist us with and make available for us. And then we have, under the Diplomics banner, we have Jacques, who's taking care of all the videos and all the promotion and marketing. So there will be videos of today. These will be available online for you to also share at your schools and your foundations. And then to Mhlekazi, who's assisted with all the administration parts, and to Anya at the back, who likes to hide, but she will be assisting in the lab as well. And then to my team at we're at the Institute for Microbial Biotechnology and Metagenomics. So we have Dr. Mpumi, say hi over there, and then a very enormous, huge thank you to Dr. Leon Mark. Give us a wave. So most of you have interacted with her. She has actually taken full responsibility of this workshop, and everything that's happening today is through her own hand. So she's no longer with me. She was a postdoctoral fellow up until the end of March and sadly left us. We miss her, but thank you, Leonie, for everything that you have done and for continuing to see this program through, even though you're no longer employed by us. And then the trainers today, we have Stephanie Lawrence over there. We have Dr. Lonnie van Sale, who's quickly run back to the lab to fetch the sequencer. And Chelsea Aries over there, together with Anya. So they will be giving you the actual training later this afternoon. All right, so I also want to introduce the speakers. As I said, the program is very tight. Two of them are joining us online through remotely. So I want to introduce them now. Gwyneth is actually in the screen over there. So Dr. Gwyneth Matches, she's at the South African Institute for Aquatic Biodiversity. This is a national research foundation. What do you call your Gwen?
Business unit.
A unit.
Yeah.
But this is based at Rhodes University. So Gwen, she's an instrument scientist at Rhodes, where she conducts research related to molecular ecology, genomics, and environmental DNA studies. And she also provides support and access to infrastructure for researchers wanting to undertake genomics research. So she'll be our first speaker. I'll get to you in a second, Gwen. Then we have Yiting Ho, who's in the audience with us today. So she's from Anchor Environmental Consultants. So we try to bring academic and also industry to, exposure to this workshop. So Yiting is a marine biologist with expertise in genetics, biostatistics, and scientific diving. She's proficient in next-generation sequencing with extensive experience in biological sampling, and her scientific diving skills and biostatistical proficiency complement her ability to analyze and interpret complex ecological data effectively. So we hope to, already she's shared that she's going to talk a bit about her master's work. No? Oh, all right, excellent. So I'm really looking forward to your presentation. And then we have Dr. Rob Finn. So he's at the European Molecular Biology Laboratory in the European Bioinformatics Institute in the UK. He is the co-chair of the BlueBiobankBiotech project and a collaborator of ours. He co-leads the project. He also leads EMBL EBI's sequence families team. Everything bioinformatics in science is done because of this group. So they, to me, are rock stars, and they have completely made a huge service to science worldwide. So they develop a lot of the platforms and the bioinformatics tools that we use for genomics and all the omics research. So yes, we're looking forward to Rob presenting later today. All right, then just a little bit about us, the Institute. We're at the University of the Western Cape. Has anybody been there? Do you know where it is? Yes? Okay, so we're in this beautiful building. Our institute was launched in 2007. I'm the director of the Institute since 2012. These are our beautiful facilities, our laboratories. We love having visitors at UWC because they never expect to see this. The Institute has 8 staff members and 40 postgraduate students. And what do we do? So this is a very big slide, but we are microbiologists. So we only focus on microbiology, and the intention is to access microbial diversity from a very wide range of environments from all over the world, in the hope that we can study them, but also make something that is valuable to humankind. Our projects feature in all of the biotechnology sectors. So we have projects that look at microorganisms that can be used to develop agricultural solutions, for human health, for industrial application, and wastewater treatment in the environmental sector. So we do a lot of very diverse research projects. We use a very wide range of technologies, and we have very cool facilities and infrastructure at our institute that is quite rare in a single academic laboratory in South Africa. So we're very well equipped. We try to bridge the fundamental research with the applied and commercialization of our research. And so we collaborate very extensively with academic and industry partners throughout the world. And we have a number of service platforms at the Institute. Genomics is one of or next generation sequencing is one of these service platforms that we offer to the research community in South Africa. As I said, our approach is to try and access microbial diversity in a wide range of environments. And I don't know if I saw one of the questions was to see what your understanding is of metagenomics. And that's because the whole field of microbiology is actually very limited in what we understand, because we can only culture less than 1% of any microorganism in a laboratory. And so we develop and use a variety of approaches to be able to study our microorganisms and look at what they're capable of producing. So we typically start at some environment, somewhere exotic sometimes, somewhere not so exotic. And we do a classic bacterial isolation. I assume this is the kind of thing that you teach your students. We then, screen these cultured organisms for some kind of activity that we're interested in, and then we follow, either isolating an enzyme or isolating a compound that we can then apply in the industrial process or industrial application that we're interested in. And this is generally what is known about microbiology. But in the late 1990s, early 2000s, became realized that actually we cannot study the majority of bacteria in the laboratory, because we cannot replicate the kind of conditions that they are familiar with in a laboratory. And so we're actually missing out on the majority. And so that's where the field metagenomics was born, where tools and technologies have been developed over time where we can still access the functional capacity of these microorganisms without ever having to culture them. And so this is really something that has exploded over the last 20 years, and that's essentially where omics, I think, really comes from, is using omic technologies to still conduct this kind of work, but without having to culture the organisms. And so Sequencing is a very big component of this. We also have different ways of manipulating the genomes to still be able to screen for the activities of the compounds or enzymes that we are interested in. So that's our logo, putting microbes to work. And the idea is that if we can access environmental diversity, so different environmental factors will have a different impact on the microorganisms and the way that they evolve and adapt to those environments. And so that breeds new or different or unique microbial diversity, and hopefully that also leads to biochemical functional diversity, which we then access at the end of the day. Because we have sampled from so many, this is just a snapshot of some of the environments we've been. As you see, it's been all over the world. We harbor a very large collection of microorganisms and metagenomic libraries, and there is no way that we can research and analyze everything that we have. And so we were nominated as a national biobank, the Biodiversity Biobank. This is run by SANBI, the South African Institute for Biodiversity. It's another of the Department of Science and Innovation and Technology infrastructure programs. And so at our lab, we host the National Microbial Biobank. And this is where you guys are coming in today. You're actually going to be contributing to this. So the microorganisms that you are going to be sequencing is going to be put in as they are specimens in this biobank, and so you're actually contributing to this national biorepository. Because of this, so Diplomics are running a project, a national initiative, the 1KSA project. Here the idea is to sequence 1,000 indigenous genomes from South Africa. This is plants, animals, all biodiversity. And our group are contributing 500 bacterial genomes towards this target. So we are contributing half of the targets for this program. It falls under the larger African Biogenome Project. So there's a lot of benefit to this program, both from conservation, conserving our biodiversity, but also the application of what this biodiversity can offer to science. All right, so maybe things are coming together. What the B cubed or the B3, I don't know if that became apparent, but our logo is B cubed, and that's because it's biotech, biobanks, and blue remediomics or BlueBiobankBiotech, sorry. So as I mentioned, this is being run under the auspices of the Blue Remediomics project. This is an EU-funded project that I'm involved in. It involves 24 international partners. And we're actually, I thought it would be easier if we just play a little video for you.
The marine microbiome, comprised of nearly invisible organisms, such as bacteria and algae, plays a pivotal role in ocean health. These organisms perform critical functions in the food chain, oxygen generation, and the recycling of key nutrients. The marine microbiome is also one of the fastest growing segments of the blue economy. It represents a vastly untapped resource, for example, for developing novel medicines and high-value commercial products such as enzymes. Thousands of different species of microbes have been found in the marine environment, with the vast majority not having been explored yet. To fully harness these microbes, more research is needed. Blue Remediomics will develop novel tools and approaches to explore marine microbiome data, while also working on the discovery of sustainable marine microbiome-based products, processes, and services. The project will extract knowledge from the huge volumes of DNA data collected from marine microbes worldwide. To facilitate this, Blue Remediomics is developing an open-access discovery platform to harmonise marine microbiome data along with new screening technologies to assess data for useful characteristics for commercialisation. This knowledge will be used to generate products and processes from marine bioresources, with a focus on finding new ways to reduce waste, increase the reuse of natural products, and improve aquaculture processes. As interest in the ocean's microbiome gains momentum, a key pillar of Blue Remediomics is devoted to ensuring that any discoveries also benefit the people and countries that provide them. Join BlueBiobankBiotech on its quest to explore, understand, and harness the invisible life of the ocean.
So you'll hear a little bit more about this in Rob's talk later today, but my research team is here. We're all involved in this project, so please feel free to ask us questions about what you've heard today. As I said, very tight schedule, so we're trying as best as possible to stick to schedule, but just to to remind you to just complete. I know there's a lot of forms, indemnity forms, registration forms, also for you to be able to earn your SACE point, your CPD points. Please complete the forms that are circulating. And I think that's it from me. So again, welcome. It's wonderful to have you, and we're really excited to work with you through the day.